The final glucose degrading pathway that we're going to talk about is called the Ettner-Duderow pathway, and this pathway is an alternative to glycolysis or Ebden-Meyerhof pathway. I like to think of it as the two-in-one shampoo because it actually produces products that both the penose phosphate pathway would produce and that the Ebden-Meyerhof pathway would produce, but it is really just an alternative to the Ebden-Meyerhof pathway. We see it mostly in soil bacteria, so some of the bacteria that will appeal to Eric with his interest in bioremediation, for example, Pseudomonas, Rhizobium, Azotobacter, Agrobacterium. A very big environmental consequence are some of these microorganisms. So these are the ones in which the Ettner-Duderow pathway has been most noteworthy and celebrated. They're not actually the only bacteria that run the Ettner-Duderow pathway. There are a few other gram-negatives. Mostly gram-positives don't use this pathway, but there are a few. Those of you who had unknown number 150, Enterococcus faecalis, that actually is a bacterium that I like to call it a duck out of water because it's a gram-positive that has certain behaviors like a gram-negative, and one of those is that it does utilize the Ettner-Duderow pathway. So again, this is an alternative glucose degrading pathway, something that these bacteria have instead of the Ebden-Meyerhof pathway. So the steps on this fit that two-in-one shampoo idea. That is, the trio stages are identical to those that we see in the Ebden-Meyerhof pathway. So you're going to recognize all of those stages where glyceraldehyde 3-phosphate is further oxidized, forming pyruvate. You've seen these stages before, so those will be recognizable. And then likewise, the hexo stages are very much the same as what we see in the penose phosphate pathway, or at least very similar. So we're still going to see that sort of oxidative nature of the first stage of the penose phosphate pathway making itself known in the first stages of the Ettner-Duderow pathway as well. Let's take a look at this pathway. It's really, it's quite a unique one and very cool. Looking at some of the things that we recognize from the penose phosphate pathway happening first, and in fact the same enzyme converting glucose 6-phosphate to 6-phosphogluconate. We didn't name that intermediate in the penose phosphate pathway, but that is the first intermediate and of the first oxidation in which we get the production of NADPH. So notice that unlike the Ebden-Meyerhof pathway, unlike traditional glycolysis, the Ettner-Duderow pathway does produce NADPH. So this pathway can support a certain amount of need for reductive biosynthesis, producing NADPH where Ebden-Meyerhof does not. The kind of quintessential or key intermediate in this pathway is 3-keto, 3-deoxy, 6-phosphogluconate. It's quite a mouthful. And it is split down into the 3-carbon intermediates. But notice that rather than being split into 2 molecules of glyceraldehyde 3-phosphate, like we see in glycolysis, it's split into 1 pyruvate and 1 glyceraldehyde 3-phosphate. That means that the stoichiometry on that from here out is 1. So note that this pyruvate's all done. So now only 1 molecule of glyceraldehyde 3-phosphate is oxidized in the steps that you now recognize, getting converted to big old 1,3-BPG, worth its weight in gold and able to make ATP, 3-phosphoglycerate and 2-phosphoglycerate, and finally our good friend PEP or phosphoenolpyruvate. So we see these being the steps that we already know in which NADH is generated, ATP is generated, and ATP is generated again to give a net yield of ATP. So let's see if we can challenge our minds to determine the net yield of the Ettner-Duderow pathway, because it's definitely different than the Ebden-Meyerhof pathway. So let's kind of work our way through this, noticing that we get the input of an ATP in step 1. That's not unfamiliar because this is the exact same step that we see in glycolysis. So -1 ATP. But here we get in step 2 +1 NADPH. That's just not something we saw being produced at all in glycolysis. And then we ramp through these remaining steps, but at a stoichiometry of 1 for the trio stages, we get +1 NADH. And now we're at a net of 0 ATP because we've invested 1 ATP up here and net 1 here, so we're at 0. And then finally +1 ATP down here in the final step, in which PEP gets converted to pyruvate, very recognizable step for us. So notice then, if we were to write the net yield, overall yield for this pathway, we've got 1 NADPH, 1 NADH, and 1 ATP for, again, a 1-to-1-to-1 stoichiometry, 1 NADH, 1 NADPH, 1 ATP. And that's the overall net yield of our 2-in-1 shampoo pathway, making all of the types, or making 2 types of reducing power and ATP at the same time. Most bacteria that have the Ettner-Duderow pathway also run the pentose phosphate pathway, but they don't run glycolysis. So here's where they're getting their ATP, their NADH, they're getting additional reducing power in the form of NADPH to run biosyntheses. So many soil microbes and some others are going to really love this 2-in-1 shampoo pathway. So moving along from here, I want to make sure that we revisit the fates of pyruvate. So remember that in our canyoneering analogy, we said that once pyruvate is formed, which we know to be the product, whether we're talking about the pentose phosphate pathway, the Ebden-Meyerhof pathway, or the Ettner-Duderow pathway, our goal now is to say, well, what might happen to that pyruvate? There are many possible things that could occur depending upon what's going on in the cell. So we now get to entertain these many fates of pyruvate, choosing which direction it goes based upon the needs of the cell. So let's begin by thinking about the possibility that pyruvate could get further oxidized. So in a yeast cell, as my cartoon shows, that would occur in the mitochondrial matrix. These 2 pyruvates would go into the matrix where they would be further oxidized, remember, forming the 2-carbon acetyl-CoA groups, and also concomitantly producing that thoroughly worn out, fully used up carbon dioxide, because there's a carbon lost here going from a 3-carbon molecule to a 2-carbon molecule. And also remember that this is a redox reaction where more reducing power is produced. So NAD plus is reduced to NADH. And of course we have a taxicab cofactor added on here in the form of coenzyme A, and we'll talk more about that later. So this is one possible fate of pyruvate, and I'm just going to label this fate number 1. But again, it's not the only thing that could happen to pyruvate. And we know certainly that pyruvate provides a lovely 3-carbon LEGO that could be used to build other things. For example, pyruvate could simply get used as a building block to build the amino acid alanine. This is a good example of the use of pyruvate as a precursor metabolite. So fate number 2, I'm going to draw in here that this is 2 indicates that pyruvate could be used as a precursor metabolite, or a Lego to build bigger things. The third possible fate of pyruvate comes along when the possibility of further oxidation is gone. And remember how we talked about that before. For example, with the yeast, when we put the stopper in the bottle and we starve the yeast of oxygen, their terminal electron acceptor, we basically say, hey, you can't use your ETC anymore. The TCA stops. All of the jazz happening in the mitochondrial matrix comes to a screeching halt. And the only possibility is that pyruvate could get used itself as an electron acceptor. So pyruvate, or a derivative of pyruvate, can get used to just simply recycle the NADH that's made in step 6 of glycolysis. And that enables glycolysis to keep on running. Remember, this fate that I'm describing, 3, is the use of pyruvate or a derivative of pyruvate in fermentation. So this is fermentation. Of course, we know that within a yeast cell, that fermentation is totes going to be alcoholic, right? So our product of this kind of fermentation is going to be ethanol. And we're going to talk a little bit more very quickly here about this third possible fate. So reminding ourselves of these three possible fates for pyruvate, beginning with the idea of further oxidation, that pyruvate gets ripped and stripped further of its high energy electrons in the transition step and the TCA cycle, and the electrons, the reducing power formed, goes to the ETC and makes more ATP, right? That's the high energetic, super amazing yield fate of pyruvate. But we know too, pyruvate could get used as a precursor metabolite, the Lego, to build other molecules. And then, of course, remember that our third and maybe our most favorite, depending upon the day, fate of pyruvate is the use of it or a derivative of it as an acceptor in fermentation. So this allows for the production of these incredibly useful products, such as the fermented sourdough bread, yogurt, sour cream, buttermilk, beer, wine, and other alcoholic beverages. So all of these amazing products that we often celebrate will see being a product of this fate of pyruvate. So let's jump in with alcoholic fermentation. And of course, all of you know that yeast are the most quintessential example of a fungus that utilizes alcoholic fermentation. But what you may not have known is that there certainly are also some bacteria that produce alcoholic, that perform alcoholic producing fermentation. And I even read an article a while back about a corporation, a nascent corporation, that was trying to utilize bacteria to make booze as an alternative due to ease of production. So next thing you know, we'll have bacteria beer. But then also proteins can sometimes utilize this type of fermentation. So the long of the short is that here we have glycolysis in a nutshell, where we remember that the yield of glycolysis, or Ebden-Meyerhof, was 2 NADHs and 2 ATPs. Well, the 2 ATPs are always going to be worth ATPs, but remember that the NADHs are only going to be worth ATPs if there's a functional electron transport chain. Of course, if there is no functional electronic transport chain, or that electron transport chain has no terminal electron acceptor, then we know that the process of NADHs getting to cash in there for ATPs is stopped. So what's going to occur in alcoholic fermentation is that pyruvate is going to get decarboxylated and converted into a molecule called acetaldehyde. That's what I mean when I say a derivative of pyruvate. Acetaldehyde is a derivative of pyruvate. So now this derivative of pyruvate simply has the job of accepting the NADH electrons that were produced in glycolysis, because in the absence of a place to take those electrons, these NADHs are absolutely worthless. So now acetaldehyde says, you know, I'll take one for the team, right? I'll take on those electrons so that the glycolytic pathway can keep on going and keep on making ATPs. So when acetaldehyde gets reduced by those two electrons, you got it, ethanol, right? The yeast begin to make alcohol. So it's tempting to think that the goal of alcoholic fermentation is to make booze because we like the byproduct. You know, it's tempting to think, oh yeah, those crazy yeast, they just want to get drunk, right? And that's why they make ethanol as a byproduct for their fermentation. But that's not the case at all. In fact, ethanol, as some of you will attest to, in excessive amounts is toxic. And if it accumulates to a high concentration, it actually is a toxic byproduct that eventually kills the yeast. So the yeast will actually die due to the production of ethanol. So the point for them is not to make alcohol. That's not the point of doing this. The point for them is to regenerate NAD+. So really, if we want to highlight an objective for the yeast cells, it's to make more NAD+ to enable step 6 of glycolysis to keep on going, to allow them to keep on making the 2 ATPs that they are making per glucose. So what happens is this is basically a short little cycle allowing for the regeneration of NAD+ and the continued production of ATP. This is also the goal in other kinds of fermentations. And let me tell you, there are many types of fermentation, alcoholic only being one of them. And of course, our food lovers in the group are going to tell us, and I think Kelsey, maybe Haley, some of you had expressed a love of cooking and of food, good food, and so much good food comes from lactic acid fermentation. In fact, our probiotic bacteria of our microbiome, the lactic acid bacteria, are some of the most important. So the lab that we've talked about many times before, I know I've had multiple conversations with Brandon about the lab, the lactic acid bacteria produce lactic acid as a result of fermentation. Now, what's different between them and yeast is that most of the lactic acid bacteria completely lack an electron transport chain. So it's not that they get starved of their terminal electron acceptor the way that yeast do, but what instead is happening is that they never had the option of further oxidation for pyruvate. That is, their pyruvate molecules don't even have that possible fate available to them. So these bacteria completely lack an ETC. So the only route for the fate of pyruvate to generate ATP is through fermentation. And it's slightly simpler than alcoholic fermentation because the pyruvate, in fact, gets utilized itself rather than getting converted into a derivative. It gets utilized itself as an electron acceptor for that NADH to regenerate the NAD+. So this forms the lactic acid. Now, as Sawyer will attest, when he's running 400-meter intervals, the lactic acid bacteria are not the only organisms that utilize lactic acid fermentation. When we run out of terminal electron acceptor in the form of oxygen, we too will meet some of our energy needs anaerobically using lactic acid fermentation. There are also proteins that do this. Algae, for example, chlorella, which is an edible algae. In fact, if you like multi-green kombucha, you will find chlorella being one of the ingredients in that. So other organisms besides just the lactic acid bacteria use lactic acid fermentation. And in fact, some organisms use lactic acid fermentation and alcoholic fermentation. I mean, they're the fun guys, right? That will enable both the production of lactic acid and alcohol from their fermentations. These are called heterolactic fermenters, meaning hetero meaning more than one. So they produce more than one product of fermentation, whereas the homolactic fermenters produce only lactic acid. So these are the two types of fermentation that right now we're covering in detail. You can look forward later to covering other types of fermentation in lab. But I do want to make mention that no matter the type of fermentation, the goal for the organism is always regenerating the NAD+ to keep glycolysis moving, right? The point is to keep that glycolysis making those ATPs in dire circumstances when that is the only way to get energy. So the purpose of fermentation is to regenerate the NAD+, to keep glycolysis going, to keep the movement of ATP production, even in the absence of an externally derived electron acceptor. Often that externally derived electron acceptor is oxygen. So a bit of terminology clarification there. So when we say an externally derived electron acceptor, we're talking about a terminal electron acceptor for the ETC. For us, that's oxygen. For yeast, that's oxygen. We're going to learn about some bacteria that use things like sulfate as a terminal electron acceptor. So producing things like hydrogen sulfide as their byproduct. Remember how we breathe out CO2 and water being the product of reducing oxygen as our terminal electron acceptor. Well, those bacteria that use sulfate as a terminal electron acceptor, they breathe out hydrogen sulfide gas, right? The smelly rotten egg kind of smelly stuff. So depending upon what that externally derived electron acceptor is, will dictate that product. So in the absence of that externally derived electron acceptor, a cell has to use an internal electron acceptor, pyruvate or a derivative of pyruvate, in dire straits in order to keep on making that ATP. Or it's just its lifestyle, right? Like the lactic acid bacteria. Let's look at a quick table or graphic of different kinds of fermentation, looking at the two that we've covered in detail first, alcoholic fermentation where pyruvate is converted to acetaldehyde and then to ethanol, of course, with the goal of regenerating the NAD+. Pyruvate converted to lactate in lactic acid fermentation, same goal. But there's also propionate as the formation of the product, the acidic byproduct that propionobacteria will make. So in fact, if you like Swiss cheese, and I don't know anyone else is like me, at an older age, I started craving Swiss cheese. I didn't like it when I was younger, so kind of an acquired taste. The Swiss cheese is made with propionic acid fermentation. Now, certain bacteria that we work with in lab, such as E. coli, can utilize what's called mixed acid fermentation. So certain of our Enterobacteriaceae, enteric bacteria, will use that fermentation, making a lot of different mixed acid and or alcohols and things like that. 2,3-butanediol is another possible product for other kinds of Enterobacteriaceae. For example, Enterobacter and Klebsiella will make that as a byproduct of their fermentations. So sometimes which kind of fermentation is used by an organism helps us to identify that bacteria. So all the fermentations, many different fermentations, and yet the goal is always the regeneration of NAD+ to keep glycolysis kicking and moving.